good. Okay, let's start. Good afternoon, everyone. My name is Lander Fu, and please let me introduce you our company, ReadCrystal, a CRO company specialized in X-ray crystallography, cryo-EM, MicroED, and fragment-based drug discovery. And I will share our distinct advantages in accelerating theoretical research and new drug discovery. First, I would like to share how our company started. In 2018, MicroED was awarded of the top 10 global technical advancement by science, and it is a novel technology in structural determination. Our CEO, Dr. Leifen Liu, and the scientific advisor, Dr. Junliang Sun, were in the research team which invented this technology in Stockholm University in Sweden. And they later started ReadCrystal, and now we are the first MicroED platform and the leading company right now. Now, ReadCrystal Biotechnology is a CRO company specializing in structural analysis service. We have over 50 people. The professional research team and high-end instruments allow us to provide the best services to our clients and obtain the best results. So far, we have collaborated with over 350 partners and have completed over 750 projects. Many of them are published on journal, and some of them are used in patents. Here are some pictures of our laboratories. First one is the electron microscope lab. We own a 200 kilovolts EM, and the following is the biology lab. We do general experiments and purification there, and the crystallization lab. We can do screening and grow crystal there, and the following is the solid state characterization lab. Last year, we obtained the CMS certification, which proves we are able to provide the verified result for our experiments. The next page is collaborating synchrotron for X-ray crystallography. The most common one we use is Shanghai Synchrotron Radiation Statistic, SSRF, located in Shanghai. It is very close to us. We have a beam time every 2 weeks, and we are able to send the sample within 3 hours. This shortens the waiting time and minimizes the damage during shipment. Also, we can ship our sample to other synchrotrons worldwide if we need or our clients request. For example, diamond in Europe, Spirit in Japan, and Australian synchrotron, which is in Australia. In the next section, I will go through details of our services, followed by some case studies. From pioneering MicroED innovators to fast-growing structural biology CRO partner, now our services including protein production, expression, and purification, X-ray crystallography, cryo-EM, SPA, MicroED for structural determinations, and fragment-based drug discovery. First, protein production. ReadCrystal provides one-stop solution, which means our clients can just send us a string of a protein sequence to express and purify the protein for the following experiments. Our clients can send their samples too if they like. Our professional production team will design the experiments and obtain samples in high time efficiency and purity. The next, X-ray crystallography. It is a solid method to determine protein structures, and it has been developed over 60 years. It is ideal for any protein that can crystallize, like kinase, GPTs, antibodies, enzymes, ligand protein complexes, peptides, and more. We will optimize the crystallization over thousands of conditions. And once we obtain the crystal, we immediately send them to synchrotron to collect diffraction data, and then process them automatically and manually to get 3D protein structures. The whole process usually takes 1 to 2 months. The lots of time consumed is to grow a perfect crystal, and the difficulties varies depending on proteins. Okay, here is a simple simple case of KRAS G12D, which is how target related to cancer. We did expression and the purification ourselves, and then grew the crystal. And finally, we solve a 1.7 angstrom structure, clearly showing its cofactor GPPCP. And you can also clearly see the electron density map. This one is the cryo-EM single particle analysis, SPA. This method got a great leap several years ago, and now we can use it to obtain high resolution structure without growing a crystal. It is a very good choice for those proteins which are very hard or even impossible to crystallize, especially for those large and multi-domain proteins, various shell proteins, and a very hot topic recent years, membrane proteins. We have sufficient EM assignments with our partners. Once we prepare the samples, we go quick testing on the negative string, and then we do initial screening with 200 kilovolts EM and go for 300 kilovolts EM to collect high resolution data. Finally, we reconstruct and analyze the 3D structures. The whole process usually takes 1 to 2 months. Here is a case study on memory protein GCGR. Its result was published on Science. From this 2D classification pictures, you can see we dissolve the protein with the detergents, and the 2D classification successfully gave the classes. And we obtained a 3D and 3.7 angstrom structure, which is already very good for memory protein. And you can also see some interactions between amino acids. The next part, MicroED. Instead of using X-ray to collect diffraction data, we use electron beams. Their mechanisms are similar, but electron beams are much more powerful. ReadCrystal has many successes in using MicroED's unique advantages. First, to do X-ray diffraction, you have to have a large crystal over 50 micrometers in size. It consumes more samples and it requires very high purity because the impurity will heavily affect the X-ray diffraction. And there's no way to obtain information on its absolute configurations. On contrast, MicroED uses nanocrystal in size of 100 nanometer, and it only requires a very little of samples. It accepts sample with only 1% of purity. Basically, we can just pick up samples crystal under electron microscope and do the experiments. And also, with ReadCrystal PED technology, we are able to solve absolute configuration, which is very important for some new drugs. Here are some pictures of hardware and software we use. ReadCrystal engineers customize the 200 kilovolts EM for MicroED. And with its self-developed software, we're able to screen and collect the 200 nanocrystal at one time. So how we do MicroED? First, we need some nanocrystals. You can grow the crystal yourself, or you can use the drug powder because they are already nanocrystals. Or you may just mash the drug pills into nanocrystal. Then we collect the electron diffraction data and solve the structure, similar to X-ray diffraction. The typical workflow takes only 1 to 2 weeks because we can collect the data ourselves downstairs. There's no waiting time. The method can be used on determining the structures of drug molecules, product, and some natural products from plants and bacteria, and so on. Here's a case study on oseltamivir phosphate. It is a leading anti-influenza drug, and it cannot grow large crystal for X-ray diffraction, so you have to have another way. ReadCrystal PED technology, precision electron diffraction, successfully resolved this structure, including its absolute configuration. We have solved the oseltamivir structure for many companies, including Japanese companies. Their results were already acknowledged by FDA and CDE. Here you can see the the structure of this drug. The next, we can also use MicroED on new materials. For example, nanocrystals, coughs, morphs, or minerals. The principles are very similar to drug solid state. Here is the case that our research team collaborating with MIT team to resolve a morph structure. Those are very hot topics in the new material area. Okay. And the final one is FBDD. Because we have all those experiment methods, we are able to do FBDD on our own. So first we produce the protein, and next we do a parameter screening with some experiment method and assay like SPR, TRIC, ASMS, or nano DASM. Once we obtain the positive results, we further analyze them with heated validation, and we can solve the structures to see the exact binding mode by all those methods I mentioned before. This process can takes half years. Okay. So until today, we have over 2,700 fragment base, and we have all those experiment assays and assays, experiments and assays on our own. So we can do this very quick time manner. So okay, that's it. So here I show some pictures of our partners and clients. So most of them are in China, but we are also collaborating with research groups and industrial customers from Japan, Europe, and North America. So if you are interested in our service, or if you have any questions, please contact us with the following information. Our company is located in Suzhou, a beautiful city near Shanghai. I recommend you to take a visit to the city and to our company. Thank you.